So now let's go ahead and get into the visual system. And this section is a little bit longer, so unfortunately this is probably going to go into next week. So we'll try to cover as much ground as we possibly can. So these are all the major things we're going to cover in vision. We're going to talk about emetropia, which is normal vision, versus ametropia. These words are so weird because these sound so much alike. Emetropia versus ametropia. So it's kind of like adduction versus abduction. Yeah, it's super annoying. The anatomy of the eye, which just basic anatomy. Some of the major things that we're going to go into are like the fovea, macula, versus like the optic disc. So we'll talk about some of those major structures. Pathophysiology behind macular degeneration, which is unfortunately a pretty common age-related degeneration of vision. Horrible stuff. You lose basically your central field of vision. We're going to talk about different cell types, cones. We're going to talk about the rods and all the other cells that are part of the retina. We're going to talk about phototransduction. We're going to talk about color perception, color blindness, on and off pathways, retinogeniculo-cortical pathway of vision, et cetera, et cetera, et cetera. So good stuff. But to begin with, I really wanted to talk about synesthesia because I think synesthesia is one of the most interesting conditions in neurology. Synesthesia or synesthetes, these are individuals that basically have like crossing over of sensory input. These are people that can hear colors, or they can when they hear a sound, or they can see colors in like numbers, right? So they get like crossing over of sensory input. They could taste colors sometimes, right? So there's color synesthesia. They can experience this in the inner mind, or they can also experience it in the outer world, right? Where they're like projecting that to the outer world. So some people will see like numbers, and then they'll like kind of see that as being represented by colors. So super cool stuff. It has to do with the posterior superior parietal lobe, and it also integrates like the visual, auditory, motor cortex as well. So let's watch a video on this because I think it's super fascinating. By the way, before I begin, does anybody here know any synesthetes?
Okay, the only thing that I would say is like because there's like a trend that would happen a little while ago where like people were talking about this kind of thing.
Yeah.
It was like everybody has like certain versions of this. So like what's the difference between like, this is just a random example, like someone says like Wednesday is purple, stuff like that. Like what is the difference between that and like this actual condition?
Yeah, that's hard to tell. It's really hard to say because there's like people who are like true synesthetes where they literally will taste a color or they'll taste a sound.
Like, oh, like, is this color like even or odd?
Yeah.
I don't think that would necessarily fall under the category of actual synesthesia. These are people that actually physically perceive it, right? It's like a sensory perception. It's not just like, oh, that song is like kind of blue,
Okay.
It's like a blues song. I don't know.
No,
Yeah.
that's literally an example.
Yeah. Yeah, it's more like they hear the song and they actually see blue. They see it.
Imagine a world in which you see numbers and letters as color, even though they're printed in black, in which music or voices trigger a swirl of moving colored shapes, in which words and names fill your mouth with unusual flavors. Jail tastes like cold, hard bacon, while Derek tastes like earwax. Welcome to synesthesia, the neurological phenomenon that couples two or more senses in 4% of the population. A synesthete might not only hear my voice, but also see it, taste it, or feel it as a physical touch. Sharing the same root with anesthesia, meaning no sensation, synesthesia means joined sensation. Having one type, such as colored hearing, gives you a 50% chance of having a second, third, or fourth type. 1 in 90 among us experience graphemes, the written elements of language like letters, numerals, and punctuation marks, as saturated with color. Some even have gender or personality. For Gail, 3 is athletic and sporty. 9 is a vain, elitist girl. By contrast, the sound units of language, or phonemes, trigger synesthetic tastes. For James, college tastes like sausage, as does message in similar words with the H ending. Synesthesia is a trait, like having blue eyes, rather than a disorder, because there's nothing wrong. In fact, all the extra hooks endow synesthetes with superior memories. For example, a girl runs into someone she met long ago. Let's see, she had a green name. These are green. Deborah, Darby, Dorothy, Denise. Yes, her name is Denise. Once established in childhood, pairings remain fixed for life. Synesthetes inherit a biological propensity for hyperconnecting brain neurons, but then must be exposed to cultural artifacts such as calendars, food names, and alphabets. The amazing thing is that a single nucleotide change in the sequence of one's DNA alters perception. In this way, synesthesia provides a path to understanding subjective differences, how two people can see the same thing differently. Take Sean, who prefers blue-tasting foods such as milk, oranges, and spinach. The gene heightens normally occurring connections between the taste area in his frontal lobe and the color area further back. But suppose in someone else that the gene acted in non-sensory areas, you then have the ability to link seemingly unrelated things, which is the definition of metaphor, seeing the similar in the dissimilar. Not surprisingly, synesthesia is more common in artists who excel at making metaphors, like novelist Vladimir Nabokov, painter David Hockney, and composers Billy Joel and Lady Gaga. But why do the rest of us non-synesthetes understand metaphors like sharp cheese or sweet person? It so happens that sight, sound, and movement already map to one another so closely that even bad ventriloquists convince us that the dummy is talking. Movies likewise convince us that the sound is coming from the actors' mouths rather than surrounding speakers. So, inwardly, we're all synesthetes, outwardly unaware of the perceptual couplings happening all the time. Crosstalk in the brain is the rule, not the exception. And that sounds like a sweet deal to me.
That was super poetic. It's a very good one. So that's synesthesia. Really cool condition. It's not a disease. All right. So here's visible light. This is the stuff that we can see, right? Visible light falls within a certain range, 380 to 750 nanometers. And that's part of the electromagnetic spectrum. The electromagnetic spectrum encompasses all of the radiation, going all the way from long wavelength radiation all the way to really, really short wavelength radiation. So gamma rays especially are going to be really, really rapid, right? Some of these are going to be ionizing, right? As soon as you get past ultraviolet and beyond is ionizing radiation, right? There's ultraviolet, you got UVA, you got UVB. UVA, not as bad, but UVB is the bad one. We say UVB is bad, right? I don't know. And that can cause like skin cancer, for example. X-rays, they're going to be a little bit more rapid. Those are also ionizing. They can cause cancer. If you get exposed to a lot of X-rays throughout your life, you're going to have a higher rate of developing cancer. Radiologists, they wear a lead vest. They have a lead, almost like a little scarf that covers their thyroid gland. Right? It's because they get exposed to a lot of ionizing radiation just as part of their job. And then when you get past visible light, when you get into the red spectrum, that's when you start getting into infrared. That's going to be transmitted by things like heat, fire. Then you get into microwaves, right? That helps to increase the speed of molecules that are more like liquid, right? That's why solid things have a hard time of getting hot in a microwave, whereas liquids are much more easier to heat up in a microwave. And then you start getting into like long wavelength. So there's like AM, FM radio wavelengths. There's also broadcasting type wavelengths for TV and stuff like that. Pretty cool stuff. You know, visible light though is a very, very, very narrow spectrum of all this. Are there animals out there that can perceive other types of colors that are beyond our visible range? Absolutely. For example, butterflies, hymenopteras, they perceive things in ultraviolet light. And that's kind of what helps them like see food sources, aka flowers, right? They perceive it in a different type of color. Some animals perceive things in infrared. Mosquitoes perceive in infrared, right? When they see something that's warm, they're like, oh, that's a meal. I'm going to go fly to it and, bite it, right? And so different animals are able to perceive in different spectrums on the electromagnetic spectrum. So pretty cool stuff. Light is going to be emitted from the source. Once it hits an object, the object is going to reflect the light. The wavelength corresponding to the color of the object we see is reflected back at us. All the other wavelengths are going to be absorbed within that object. So we're only really seeing what's being reflected. Now, in terms of the anatomy of the eye, this is what the eye is made up of. You have the retina, which is going to be where you have the innermost area, right? So that's going to be the sensory regions, right? That's what perceives those photons, right? Photons hit the retina, and then that gets transmitted into spikes, right? Neuronal signaling. You also have your ciliary body. The ciliary body is what allows for constriction, right? So you allow for changing the shape and size of the lens. Not the size, but the shape, right? So it can either make it more long or it can make it shorter, right? Depending on what you're trying to focus on. Light is going to be entering through the cornea. Then it's going to hit the aqueous humor, which is in the front portion of the eye. Then it's going to go through the lens, and then it's going to go through the vitreous humor, which is in the main body of the eye, and then eventually it's going to hit the retina as well. We're going to talk about the fovea in a second, but the fovea is where you have the highest concentration of cones, right? Rods, which what perceive black and white grayscale, you're going to see those especially like in other parts of the retina, but when you get to the fovea, it's going to be mostly cones which perceive color. Emetropia versus ametropia. So emetropia with an E, that's going to be normal vision, right? We all have, most of us are going to have ametropia, right? If you have ametropia, that's where you have a refractory type issue. So it's a refractive condition where the point of focus is either going to be in front of the retina or it's going to be behind the retina. And that can either cause myopia or hyperpia. With myopia, that's nearsightedness, right? Who in here is myopic? I see like half of us in here are wearing glasses. I'm presuming it's from myopia, right? If you take your glasses off, can you see far away? Not really. Yeah, I'm blind as a bat. Like when I go to get my eyes screened, I can read the top letter E. And mostly it's because I know it's E from seeing it before. And then I can barely read the second two letters at all, right? So I'm like super, super blind. So that's myopia. So it's called nearsightedness. The image is going to fall in front of the retina. Hyperpia is going to be farsightedness, and the image is going to fall behind the retina. And so you get, so this happens a lot to people as they age, right? As they age, people become more hyperpic, right? So you're going to have to start wearing reading glasses, right? There's actually a thing that happens sometimes to people who are myopic. As you get age, sometimes people with myopia, sometimes when they get older, like they don't have to wear glasses anymore. They're like, oh, my vision got better. But now they can't read. So now they have to like have reading glasses to see up close. Someone have a question? Did you have a question? No. Okay, cool. Astigmatism, this is something else that I have. So whenever I get my contacts fitted, I have to get not only my contacts for myopia, but I also have to get them specialized for astigmatism. This is where you have a distortion and blurry vision, and it has to do with the curvature of the lens. So your lens is going to be having a different type of curvature. So here's just a breakdown of everything we just talked about. Myopia versus hyperpia. Accommodation, this is where you have adjustment of the lens. Right? So the accommodation reflex is something that will allow you to either look at objects that are closer up or objects that are further away. And the pinhole effect, this is how the pupil works. The pupil has to either constrict or dilate depending on the circumstances. Right? We did this earlier on this semester where we did the accommodation reflex. We also did convergence. The accommodation reflex, which also can involve pupillary constriction and dilation, Like when you hold your finger up to somebody's face and they're looking at your finger, their pupils are going to constrict. When you look at an object far away, the pupils are going to dilate, right? So that's how the pupillary reflex occurs. And so that allows you to see things far away. You can gather more information from the environment to see objects far away. This also happens with light too. Like in darker areas, your pupils are going to dilate. So you can try to catch as many photons as possible in a dark room. So you get pupillary dilation. So here's an unaccommodated eye compared to an accommodated eye. So in this case, the accommodated eye is viewing a nearby object. And so you're going to see contraction of the ciliary muscles. Ciliary muscles are what basically allow for the lens to change its shape so that you can see objects either close up or far away. The macula is going to be the portion, like part of the center of vision. The macula is this whole area right here. So if this is a fundoscope, fundoscope is where you can like look at someone's eye either with a handheld fundoscope, or you can have a machine. If you go to like an optometrist, they have these like really awesome cameras that they're able to like look right through your eye and get an amazing picture just like this. I wish I would have kept the picture last time I got this done. It was really, I've never seen it before. I was like, wow, that's my retina. Super cool. I know Dr. Jackson has a copy of his, but yeah, super neat. This is what you see. You see a bunch of blood vessels, right? You see the optic disc. That's where cranial nerve 2 will go through, right? That's going to be where the optic nerve goes into. And then you have, oh, and by the way, this is your anatomical blind spot. We call this a scotoma, right? So when you have a blind spot, have you guys ever done that where you draw like a little dot on a piece of paper and then you move your eye away from it until the dot disappears, right? When the dot disappears, it's because that would have been otherwise perceived at the optic disc, right? That's where your optic nerve goes through. Your brain fills in that blind spot, so you don't perceive it normally, right? Your brain fills it in with whatever is like around the area, so you don't notice it. Now, in terms of the macula, again, that's like your central field of vision, right? That's also where you have the fovea. The fovea is where you have the highest concentration of cones. And then the visual field, right? That's everything that the eye can see, right? You're going to have you're going to do nasal and temporal, right? So everything in the middle, that's the nasal visual field. Things that are coming laterally are going to be picked up by the nasal visual field, right? Versus everything that's in the center of your vision, right, is going to be picked up on the temporal portion of your retina. Another thing that happens is that, as the light comes in, it crosses over, right? And everything gets inverted. So everything is backwards and upside down. And so the brain has to go through several different processes to be able to reorient and turn everything right side up again and re-invert it. So you have to have those regions of your brain re-establishing the proper orientation of objects. Macular degeneration, again, this is a really sad condition. This is where you lose your central field of vision. When you first start experiencing macular degeneration, things start looking a little squiggly in your center of vision. Like if you were to look at a grid of lines and then they crossed over, so it was like an X and Y axis type grid, in the middle, you would start seeing distortion of the lines, right? And then as it progresses more and more, you just straight up lose that region of vision. And it's really sucky because, you can't see anymore. Like you can't read a book anymore. You can't read those, the letters in a word, right? So really, really sad. There's two major types. There's wet and there's dry. Dry is going to be the most common kind. It's going to be slower onset. You get these drusen buildup. Drusens are basically metabolic wastes that start to build up. So if you did a fundoscope and you looked at the person's retina, you'd see all these like little yellow blobs basically just accumulating within the retina. And it basically causes scarring. And then you have wet macular degeneration. Wet because it involves blood vessels. And so you have neovascularization. And that basically causes bleeding into that area of the eye. And it's much more quicker onset, and you get irreversible damage.
Macular degeneration is a disease which affects the retina. In this video, we will describe the symptoms of macular degeneration.
That's a pretty long video. Actually, let's maybe
The
watch a little bit of
retina
it.
is the layer at the back of the eye
We'll watch like
which
half
helps
of it.
us to see the world around us. Macular degeneration causes changes in central vision. Images which were once clear may appear blurred. Later, dark spots may appear and enlarge. Straight lines may become distorted or curved. Colors may appear less vivid or darker. The eyes work together. When one eye loses vision, it may not be noticed because the other eye is still able to see.
So I think you guys get the picture with that one. So they're going to go into the description of dry versus wet. You can watch that video on your own time if you'd like, but for the sake of time, let's continue on. So again, here's your fundoscopic exam. So here's all of those little drusen buildup. Here's the bleed. So this would be dry for the drusen accumulation. The ones that involve bleeding, that's going to be wet macular degeneration. All right, so again, visual fields where you have projection of visual fields onto the retina. Again, everything is going to be inverted. Left and right is going to be crossed over and inverted. Up and down is going to be turned inverted as well, upside down effectively. The field of vision, you can look at the field of vision in terms of monocular versus binocular visual field. Over here, this is monocular, so you can see the individual eye and what those visual fields look like. So again, here's your nasal visual field in the middle, temporal on the sides, and then superior versus inferior. And when you cross both of these visual fields together, that's when you get that binocular type of vision, right? So a portion of it's going to be monocular, right? There's only, if you close your eye, you'll notice like which portions of your vision you're not able to perceive. Like you can't see past your nose, for example. When you close your left eye, your right eye is going to be able to perceive less because it's not going to be able to see past the nose. So things like that are going to affect your visual field as well. And then there's going to be quadrants, right? And why is that going to be important? Because we're going to talk about a couple different conditions that affect the optic tracts that are going to affect your quadrants. So you can get like hemianopsia, but you can also get quadrantinopsia or quadrantinopia, which means that one of these quadrants gets wiped out in your visual fields. So there's different conditions that can lead to that. Brain tumors, trauma, things like that. We'll talk about that. We're probably going to get to that next week, honestly. So again, here's your visual fields, right? If you follow those lines, you can see where they're going to hit the retina. So anything laterally is going to hit the nasal region of the retina. Anything that's in the medial region is going to be affecting the temporal regions of the retina. And then everything gets turned upside down as well on top of all that. So these are the types of cells that you're going to find within the retina. We already talked about these, rods and cones. Rods, black and white, right? So it's going to be grayscale. Cones, color, okay? So these are your major photoreceptors. You're also going to have a bunch of other cells that are within the retina as well. And we're going to go into all those with time here. So you have bipolar and horizontal cells. These are considered interneurons. You have amacrine cells, which are going to involve cell bodies. You also have retinal ganglion cells. Those are going to be where you have axons actually exiting out down into cranial nerve 2, right, through the optic tract. That's going to be your ganglion cells. You have glial cells, and you have Mueller cells as well. So here's, again, the structure of the retina. So if you did a cross-section, this is where the light's coming in. Notice light comes in, right? This is like in the innermost portion of the eyeball, right? Where you have like aqueous, all the aqueous fluids, the aqueous humor and all that. Light is going to come through. It's going to go through all these cell layers until it actually reaches your photoreceptors. The photoreceptors sit at the very back of the retina, right? It's going to be at the outermost area of the retina, basically. And so that's where you have your photoreceptors, rods and cones. So we're going to talk about the process of that in a little bit as well, how the electrical signals from photons, right, stimulate these photoreceptors to do their thing. Not every animal has this type of orientation. Like if you look at cephalopods, octopus, squid, right, it's interesting because they had a type of convergent evolution, right? We are totally unrelated in terms of like ancestry, right? If you did a, what do you call that? Oh my gosh, I forget what that's called. When you have like a tree of life, what's that called? What's the name for it? Oh my goodness. Oh my gosh. Where you're like tracing back like the lineage of like ancestry or relatedness of like organisms. What the heck is that called? I totally forget. It's not like Ancestry. It's kind of like Ancestry.com, sort of, but for like the animal kingdom. Dang it. It's like taxonomy. It's part of like taxonomy, like naming systems and stuff like that. And you like see like the interrelatedness of animals. Anyways, but we have like a completely different lineage of ancestry. But their eyes look so much like mammalian eyes. They're very similar. But for them, their photoreceptors are actually the first ones that get hit by the light, which is kind of interesting. So it's like inverted. Like the cell layers are going to be backwards. So it's inverted. That's considered a type of convergent evolution.
What's
So
that for? What animals?
cephalopods, so octopus and squid. So we're totally different. Like they're more closely related to like mollusks, right? And so very, very interesting in terms of how these things come about. Phototransduction. Phototransduction is how you convert those photons, right, into an actual electronic signal. And that's going to be, again, taking place at the rods and cones, your photoreceptors. They do not depolarize or produce an action potential. Right? There's going to be a whole different type of cascades taking place within this. Light activation is going to cause hyperpolarization of the transmembrane potentials. It's going to be involving channels that allow for calcium and sodium to come in, as well as potassium. And it's going to be involving cyclic GMP activation. So you have changes in the rates of neurotransmitter release. So that's going to be kind of what allows for the actual phototransduction to take place. So we'll get into that in a little bit here. So hyperpolarization of photoreceptors. When you stimulate those photoreceptors, it's going to cause hyperpolarization. So here's a flash of light on the top of the graph over here, and then you can see the membrane potential changing. So with a very minimal amount of light, you're going to see a little bit of hyperpolarization. But the more intense you flash that light, the more and more polarization you see, hyperpolarization you see taking place. Why does this hyperpolarization happen? Is because you're effectively blocking the channel through which calcium and sodium can come into. So you're basically going to hyperpolarize that cell. Right? The cell is going to become more and more negative because calcium and sodium are not entering inside. So it's a little bit different than any of the other types of cells that we talked about so far, right? With the exception of maybe like GABA type activation. So this is going to be kind of similar with like GABA, right? Where GABA activation hyperpolarizes the cell with chloride ion influx. This instead is blocking the receptor blocking the channel and preventing sodium and calcium to come in. And so in other words, when you stimulate these cells, these photoreceptors, they become hyperpolarized. They become more negatively charged. There's a couple other compounds that are going to be involved in this process. You have melanopsin, you have rhodopsin, and you have opsins. Now, rods are going to be involving rhodopsins. Cones are going to be involving just opsins. There's a few different types. I'm not going to hold you guys responsible for those. But basically, when you have light stimulation to these photoreceptors, it's going to change the conformation of these different types of opsins. So when you have stimulation of, for example, 11-cis retinol, so 11-cis retinol, upon light activation, it's going to change into all trans retinol. So it's going to change its overall conformation. And then this is going to allow for transduction. So you have light causing transduction. That's going to involve second messenger systems, PDE, and then cyclic GMP is going to be stimulated to eventually allow for closure of those sodium and calcium channels. So basically, when you have light stimulation, it just turns these things off effectively. In the dark, the opposite is going to be true. In dark, you're going to have opening of calcium channels. And in the dark, you're going to get opening calcium channels. Neurotransmitters are also going to be involved in that process as well. And I just realized we went 1 minute over class. I'm so sorry, guys. So we will continue right here next time we meet. And until then, have a wonderful weekend. Enjoy your day off tomorrow. I'm sure it's much needed for all of y'all. Last time we were talking about phototransduction, and we were discussing some of the components that you find in rods and cones. So you're going to see different types of opsins. So you have rhodopsin, you have melanopsin. Opsins are basically just specialized photosensitive compounds. It's a string of amino acids that are going to respond to photons. As you note, when light hits 11-cis retinol, it turns into 11-trans retinol. Now, you see the word retinol in there. What is that from? What's the derivative of retinol?
Retinoic acid.
Retinoic acid, which comes from where? What's there a name for that? It's you find this in the eyes, yeah. There's a certain thing that it was Easter on Sunday. What's something that bunnies enjoy eating? Carrots. What's the main vitamin you find in carrot? Yeah, vitamin A. Absolutely. That's why they say carrots are good for your eyesight. That's not a joke. It's actually true because vitamin A is in high abundance in carrots, and the opsins that you find within your photoreceptors are derivatives of vitamin A. And what was your question? You were saying something?
No, I take it back.
Yeah? All right. Sounds good. So yeah, vitamin A is going to be the foundation of these different types of opsins. And they're going to be really important in phototransduction. We're going to play a little short video in a second talking about that. So here's the cascade of phototransduction. It's going to be involving a second messenger type system. You see this in both rods as well as in cones. It's going to activate PDE, which is phosphodiesterase. Phosphodiesterase is then going to allow for the activation of cyclic GMP. And then eventually you're going to see closure of these cation channels. So both sodium and calcium are going to be blocked from entering into the cell. So in other words, you start seeing more of a depolarization type process taking place. Hyperpolarization when you block it, right? So it's hyperpolarized, you're not getting those cations entering into the cell. So that's in the exposure of light. Without light, in the dark, you're going to see the cation channels opening up. So calcium channels are going to open up. And so calcium comes in and it allows for depolarization of the cell. So this is like opposite of what we saw in normal neurons. Now, these cascades are going to allow for different neurotransmitters to be released. We're going to talk about some of the neurotransmitters. I'm probably not going to test you on them for this section on the eyes, but there's a bunch involved. There's glutamate, there's also GABA, there's glycine. There's a lot of different neurotransmitters involved in vision. I'm not going to test you on all those, but let's watch a little video talking about phototransduction.
Welcome to 2-Minute Neuroscience, where I explain neuroscience topics in 2 minutes or less. In this installment, I will discuss phototransduction. Phototransduction is the process that occurs in the retina, where light is converted into electrical signals that can be understood by the nervous system. It primarily takes place in photoreceptor cells, of which there are 2 main types, rods and cones. I will discuss phototransduction in rods, although the process is similar in cones. In the dark, positively charged sodium ions flow into rod cells through ion channels that are activated by a substance called cyclic guanosine monophosphate, or CGMP. This influx of positively charged ions causes cells to remain in a depolarized state, leading to the continuous release of the neurotransmitter glutamate. Inside the rod cell, there is a substance called rhodopsin, which is made up of a protein called opsin and a molecule called retinal, which is capable of absorbing light. When retinal absorbs light, its configuration changes, an event that prompts opsin to activate a protein called transducin. Transducin activates a type of enzyme known as a phosphodiesterase, which begins breaking down CGMP. As CGMP levels fall, the ion channels that are opened by CGMP begin to close. Thus, less sodium enters the cell, and the cell becomes hyperpolarized due to potassium ions that regularly leave the cell. Consequently, glutamate release decreases. Strangely enough, this decrease in neurotransmitter release acts as a signal that a light stimulus is present. The rod cell returns to its normal state quickly when activated rhodopsin is inactivated, and a protein called arrestin subsequently binds to it. Arrestin blocks the ability of rhodopsin to activate transducin, which makes the cascade unable to continue. A complex process then restores retinal to its original configuration, making it ready to absorb light once again.
Okay. So, interesting process. In other words, light basically turns off the cell. And again, that happens with both rods and cones. So again, here's another breakdown for this specific diagram talking about what's going on when in the dark compared to when you're in the light. So in the dark, cyclic GMP levels are going to be elevated. Those channels are going to be open, right, so that they can continually depolarize. Versus when it's in the dark, the cyclic GMP levels drop, and then all of a sudden those cation channels close up, and then you get hyperpolarization of the cell. Now, in terms of the locations of rods and cones, you're going to find rods throughout the retina with the exception of the fovea. The fovea is going to be one of those areas where you're going to see mostly cones. Okay, so that's all part of the macula. Now, we talked about macular degeneration, right? This is your central field of vision. Right? If you get destruction of this region, you're going to have difficulties in seeing like letters, right? Focusing in on specific like tiny objects, right? Also, you're going to see reduction of color because with your cones, you get color, right? This area in the fovea has the highest concentration of cones. In other words, that's where you get a lot of color perception. So in other words, this area has lots of acuity. We're going to talk about the way that rods differ from cones in terms of how many photons need to stimulate a rod versus a cone. For a cone, you need to have more photons stimulating a cone. So cones are not that sensitive to light. Rods, on the other hand, are very sensitive to light. Right? It doesn't take many photons to stimulate those rods. That's why when you're in the dark, rods are basically what take over. In light, cones, right? You're seeing color. Cones is going to be like the main thing coming into play with light. Let's watch a little video here on the fovea.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the retina. The retina contains the neural component of the eye. When light reaches the back of the eye, it enters the cellular layers of the retina. The cells of the retina that detect and respond to light, known as photoreceptors, are located at the very back of the retina. There are 2 types of photoreceptors, rods and cones. Rods allow us to see in dim light, but don't allow for the perception of color. Cones, on the other hand, allow us to perceive color under normal lighting conditions. Throughout most of the retina, rods outnumber cones. In one area called the fovea, however, there are no rods but many cones. The fovea represents the area of our retina that provides our highest acuity vision, and thus is at the center of our gaze. When light hits photoreceptors, it interacts with a molecule called photopigment, which begins a chain of events that serves to propagate the visual signal. The signal is transmitted to cells called bipolar cells, which connect photoreceptors to ganglion cells. Bipolar cells pass the signal on to ganglion cells, which leave the eye in a large cluster at an area called the optic disc. The optic disc doesn't contain any photoreceptors, and so represents an area of the retina that can't process visual information, creating a natural blind spot. However, we normally don't notice our blind spot. The brain uses information from surrounding photoreceptors and the other eye to fill in the gaps in the images that are processed by the retina. After leaving the retina, the ganglion cell fibers are called the optic nerve. The optic nerve carries visual information toward the brain to be processed. There are 2 other cell types in the retina that should be mentioned: horizontal and amacrine cells. Horizontal cells receive input from multiple photoreceptor cells. They integrate signaling from different populations of photoreceptor cells, make adjustments to the signals that will be sent to bipolar cells, and regulate activity in photoreceptor cells themselves. Amacrine cells receive signals from bipolar cells and are involved in the regulation and integration of activity in bipolar and ganglion cells.
So we're going to talk about those other cells in a little bit, the amacrine versus the horizontal cells in a little bit. Basically, what they do is they just kind of help to allow for like better vision of like contrast, right? They help to kind of like filter things a little bit better. So then the information that finally makes its way to the optic nerve will be a little bit more precise. There's one way to kind of think about that. So here's an image of a like a cross section of a retina. So down here, you notice that there's going to be your inner fovea. You're going to see mostly cones in that region. You also have rods laterally to that. Here's a graph showing the presence of rods and cones, and you can see the area that's marked as the optic disc. You're not going to have anything there, right? So in other words, the optic disc is your natural blind spot. You're not getting any sort of phototransduction taking place. That's because you have to have a location where blood vessels as well as the optic nerve are going to be traveling through. And so that's going to be within the optic disc. Also, blood vessels are going to be, there's a bunch of blood vessels in the retina, but they're going to be spread out. So it allows for light to be able to move through all the different cell layers to actually stimulate those rods and cones. Yeah.
So like in the dark, whenever we're trying to look at something, we have to look away to kind of see it.
Yes, I know what you mean.
Yeah, that would probably be because in the center field of vision,
Yeah.
cones
Like,
versus a little bit laterally is going to be more rods.
okay.
That's a very astute observation. Yeah. Yeah.
Because I know, like, whenever, like, I'm trying to, I don't know, either it's night and I'm trying to look at something in the sky, or if it's pitch black in a room, if I want to see it, I look away and then I can see it.
Huh. Yeah, that's because of the rods. Yeah, absolutely. Yeah, I never thought about that. And I never actually noticed doing that. But
Yeah.
next time I'm in the dark,
Yeah, definitely.
I'll make note of that because that's a really good observation. So yeah, that has to do with the cones. Cones are not very sensitive to light. They're not very sensitive in the dark. Now, this is one of the reasons why cones are much more sensitive in the dark. So they have a lot of convergence. So here's your rods, tons of rods, and then they're all going to communicate with one bipolar cell. So in other words, all that information is being captured by those tons of rods, which is eventually getting funneled down to one bipolar cell. So in other words, just a few photons can come in, stimulate a few of those rods, but then eventually all that information gets sent to the bipolar cell, which eventually gets sent down to the optic tract. So that's why they're much more sensitive to light within the dark, right? Versus cones, you have to have lots of photons available to be able to stimulate a cone. Now, in terms of the number of rods, it can vary, right? It can be 15 to 30 rods per bipolar cell. Versus cones, it's going to be a little bit less of convergence. Okay, moving on. Now, in terms of like the way that you describe resolution, rods, again, extremely light sensitive, but they don't have much spatial resolution. And then they're going to operate in lower light scenarios. So there's different types of visual functions. You got scotopic vision, you got mesopic vision, and then you have photopic vision. The scotopic and mesopic, that's where your rods really come into play. And so, for example, if you're in the dark and you're the only illumination you have is starlight or moonlight, that's going to fall within the mesopic visual function. Versus when you actually have daylight, right, indoor lighting versus sunlight, that's going to be in the photopic vision spectrum, and that's where cones are really going to come into play. Cones are going to have more spatial resolution, but again, they're not sensitive to low light levels, so they're going to be more of a photopic type of vision. Now, of course, if you get to a certain level of brightness, you're going to start seeing damage to the cells, right? That's why you never want to look at the sun, for example, right? I think we have a solar eclipse coming up pretty soon, right? So when there's a solar eclipse, you have to wear special glasses for that. Or you poke a hole in a piece of paper so you can see what the solar eclipse looks like on the ground as the light goes through the hole in the paper. Have you guys done that before? Yeah, pretty cool stuff. I showed that to my mom because we had a solar eclipse, I think it was last year. And I told her to do that. She never did that. She's never heard of that before. And she did it. And she was like, oh my God, it actually works. It's super cool. I can see the sun. It's like, yeah, without burning your retinas. Now, color blindness is another interesting phenomenon. This is very common. Lots of people suffer from this, especially guys. Like my best friend has this. Red-green color blindness. He can't distinguish between red or green. And this was pretty hilarious when we were we've played music together, and we started our liquor company together. And I did all the graphic design and all the artwork. And whenever we would do it together, he's like, I don't even know what the colors are that you're using right now. I'm just like, all right, well, don't worry, it looks good. So I was like, take my word for it. But when it comes to any sort of like colors, he's not really very good at that. So there's 3 different types of cones that are going to receive different types of wavelengths. So short wavelength, medium wavelength, and long wavelength. And it goes like Roy G. Biv, right? The colors of the spectrum. When you get to more like violet, that's going to be short wavelength, right? So your ultraviolet light, high, like very, very like short wavelength. Whereas when you get to the red spectrum, like infrared, that's going to be much longer wavelength. And so you have different cones that can respond to those. And that's how you get color blindness, is if you lose some of these different wavelengths. And this is the kind of vision that you might see. So this is like a generic normal vision versus red-green color blindness. And then there's other types too. So there's protanopia, there's deuteranopia, and there's tritanopia. So let's go into what those actually mean. Oh yeah, by the way, mostly affects males because the pigmentation is going to be found on the X chromosome. And guys only get one X, right? Ladies have 2 X's, so if one of those X's don't work properly, that's okay. You have the other X to make up for what's lacking. Now, protanopia is where you lose long wavelength. So that's going to be what protanopia looks like. So this is normal. You can see the flowers all nice and red. With protanopia, everything looks kind of yellowish, kind of bland, not great. Deuteranopia is loss of medium wavelength. That's what deuteranopia looks like. Tritanopia is basically the inability to distinguish between any of those colors. So on the previous slide here, that's where you see tritanopia. So everything kind of looks homogeneous almost. You can see like blues and red hues, but it's tough to distinguish between all the different color types. So that's color blindness. Also, contrast is really important in light. Now this came out like what, 2015 or so, 2014, right? So I don't know. For me, I always saw it as blue and black. Did you guys ever see it in white and green? White and gold. That's how you would perceive it when you saw the image. I always just saw it in blue and black. You can see both. Yeah, I only saw blue and black. I could never see the white and green or white and gold or whatever. It's kind of like that. The sound. I think it's Laurel versus Yanni. Do you guys remember that? Yeah, that's wild. For me, I could switch it. Yeah. You could like pay attention to it in one way and like hear it as Yanni, and then like think of it as being Laurel, and you can kind of like flip it in your mind. It's really wild. I mean, a lot of it had to do with the frequency of sound that certain people can perceive. Like, I think there was a difference between men and women when they could like perceive the different, whether it was Laurel versus Yanni, had to do with like frequency interpretation. But lighting also has a similar thing, right? So different people have different perceptions. And that has to do with contrast, as well as lighting in general. So like in this study that was released in 2015, they actually like examined like the unconscious perception of color based on whether or not the image was illuminated with warm lighting versus cool lighting. So really interesting stuff. Here's another image talking about contrast and perception. So I actually, like, when I was looking at this image, I had to zoom in and screenshot the little colors or the little boxes to see if those colors indeed matched or if they were actually different. So the one over here on the left, I even, like, in my office today I was doing this. I was like, I didn't trust this. I didn't trust the actual cube that isolated the different colors. This one, both this color and this color are exactly the same. But because of lighting and shading and contrast to the surrounding, it looks like this one is lighter than this one. But in fact, if you isolate this and do a screenshot of this brown square, and then you move that screenshot adjacent to this orange looking square, they're both brown. It's really amazing. But yeah, but with the shading, it looks like it's orange. Whereas this one over here, these are actually different colors altogether. So I actually tested this in my office before I came over here today. I did a screenshot again of this little square, and I moved it over to this square, and they were indeed different. And it all has to do with shading. So if there's any artist in this room, whenever you're like illustrating or doing like painting and stuff like that, you'll see this in your work, right? You'll have to like figure out which area is going to have different colors for like the dark hues for shading and vice versa. So pretty interesting stuff. So that has to do with contrast, shading, right? Lighting, etc. Now, the cells in your eyes are also going to help to differentiate between contrast, edge, like the edges of shapes, etc. And so there's different types of cells that come into play. There's going to be different types of ganglion cells. So you have on-center ganglion cells and off-center ganglion cells. For the on-center, if you have light within the center of a field, the on ganglion cells are going to turn on. You're going to see more spikes within those cell types. And then the off-center ganglion cells are going to turn off. You're going to see reduction in spikes until that lighting turns off. And then the off-center ganglion cells turn on. So this basically helps to allow for discrimination between different shades of lighting. That's the long story short when it comes to the on and off cells. So they respond to increases and decreases of light. In other words, changing of lighting. Again, the on-center ganglion cells, they fire when they're illuminated versus the off-center, they fire when the illumination goes down. Right? And then the way that those cells kind of basically like work together is that they allow for more definitive perception of light changes as well as contrast. Now, they're both going to be responding to glutamate. So I'm not going to test you on the different receptors for these, but the on ones are going to respond, are going to be activated when glutamate agonizes the mGlu-R6 receptor versus the off bipolar cells involve the AMPA receptor. But long story short, they're both glutamate sensitive, so they're responding to glutamate release. And this is also FYI, I'm not going to test you on this stuff, but this is you have both of these cells communicating with your like rods and cones. And so it depends on what type of lighting you're seeing. So on cells get turned on when there's lighting, off cells get turned on where there's not so much lighting. And they're going to be both inhibitory or excitatory depending on the circumstance. So the on cells are going to be inhibited by the off cells are going to be turned on by darkness. On cells are going to be turned on by lighting. Like I said, I'm not going to test you on that stuff. This is another one too that I'm like reluctant to include on the exam because these are mostly just like regulator cells, right? It's kind of like what interneurons do. You can think of these as being sort of similar to like interneurons. So they basically help to modulate signals, right? To kind of fine tune the information. So you have horizontal cells. These are going to be more inhibitory. They're involving GABA. Endocrine cells are also involving a lot of different neurotransmitters, GABA being one of them. And these are going to be lateral, right? So they extend laterally. So they're going to be communicating with multiple different cells within the retina. So again, this is just FYI. I'm not going to test you on this, but this is surround antagonism. So it's going to regulate the information that's coming from the periphery versus the central fields of vision, right? And so basically allowing for regulation of vision. And so that helps to regulate contrast, helps to regulate color, helps to regulate lighting. Here's another fun experiment using contrast, right? And contrast is a really, it's context dependent. So you have to know what the context is to be able to see the actual true color, right? So here's these little gray squares that are between the black lines are the exact same shade of gray as the gray squares within the black line. This is something else I had to do. I zoomed in, took a screenshot of those gray squares. Those gray squares are identical in their shade. But taken out of context, between the black lines, the gray squares look darker, right? Within the gray squares, because you're basically using the black as like the point of contrast or at the point of comparison, then the gray squares look a little bit lighter. Right? Same thing with these images over here with the 3 different blocks. Each of these gray squares inside are exactly the same shade. They're all the same shade of gray, every single one of them. So you can literally do this on your own if you want to go in there, take a screenshot of those gray squares and just drag them and compare them to all the other gray squares. They're exactly the same. But when taken out of context, or sorry, when put in context with the rest of the image, then those gray squares look different in color or different in shade. So. Yeah, the left side to me, I think, is even more stark in its difference, right? You're right. The right side, you can kind of see how those gray squares do look similar, but the left is like, it's remarkable how much of a difference it makes, whether it's in between the black lines or within the black lines. So there's, let's see, where are we at here? There's also layers of ganglion cells that are going to be able to interpret different aspects of an image, right? So there's going to be retinorecipient brain regions where they're actually going to form the image, but then there's the retino-no-recipient brain regions, which don't necessarily have to do with the actual image itself. What it does is it allows for like distinguishing different characteristics of the image, shading, contrast, lighting, right? So there's going to be cells specifically to recognize an image versus cells that help to specifically detail out all of those different details like contrast, shading, etc., illumination, all of that. I am not going to test you on all of the different brain regions associated with that, but there's a bunch. We are going to talk about the lateral geniculate nucleus in a little bit because that's going to be an important relaying center for vision. That's part of the retino-geniculocortical pathway, and we're going to talk about deficits within that pathway and how those deficits manifest clinically. We already talked about the superior colliculus. We're going to talk about the inferior colliculus as well when we get into audio, like hearing. Superior colliculus is going to help to relay vision as well as sound. And this is going to basically alarm your brain to something that you should pay attention to. That's what the inferior and superior colliculi do. There's the olivary nucleus within the brainstem, and then there's a suprachiasmatic nucleus as well. But all those regions are going to be involved in perception of images. So here's the retino-geniculocortical pathway. Retino, because it starts at the retina, then you have your optic nerve, which eventually makes its way down to the lateral geniculate nucleus. And again, that's kind of a relaying center for vision. So it helps to process vision as it comes in. And then eventually the vision that's processed at the LGN eventually makes its way to the primary visual cortex. Now, here are the different types of visual deficits you might get if you damage any point of the optic tract, whether you damage the optic nerve, or if you damage the optic chiasm, or if you damage the geniculate nucleus, or if you damage the pathway from the LGN to the cortex. You're going to see different types of visual field deficits. Now, recall that vision is completely inverted, right? It's totally inverted. It's not only backwards, but it's also upside down. Now, these pathways that are going from the lateral geniculate nucleus back towards the cortex, to the primary visual cortex, they're going to be flipped. The ones that are on the superior side over here are going to be interpreting the inferior visual field, right? So it's flipped, right? Versus the loop in the bottom here that's referred to as the Myers loop, that's going to be involving the superior visual field. So recall, everything is basically upside down. Now, that will make a little bit more sense when you see these types of lesions along the optic tract, along the retino-geniculocortical pathway. So these I am going to test you on. So make sure you are familiar with these different types of conditions. Now, if you completely obliterate the optic nerve, cranial nerve number 2, you're going to get unilateral vision loss in that one eye. It's going to be total vision loss, right? And by the way, let me go back real quick so you can be, where's that? So anopsia is a condition where you lose a large portion of the visual field. Hemianopsia means you're losing a portion of the visual field. So let's go back to this. So if you lose the entire one eye, that's just loss in one eye, right? Now, if you were to damage the optic chiasm, which we actually kind of talked about already when we discussed pituitary tumors way back at the beginning of the semester, where you get bitemporal hemianopsia. So remember, anopsia, you're losing a large portion of the visual field. Bitemporal hemianopsia means both of the temporal or peripheral vision fields are going to be lost, right? Remember, because those regions are what cross at the optic chiasm, right? Left homonymous hemianopsia. So that's what you would get if you have any sort of deficit on the optic tract, but it has to be unilateral, one side. So whereas in the optic chiasm, you're losing both lateral sides of the vision, if you kind of destroy the optic tract on one side of the brain, you're going to get both sides of that visual field are going to be gone. So that's why it's left homonymous hemianopsia. Now, if you start getting down into the optic radiations, right? You have those two different optic radiations. You have the ones that are the Myers loop, and you have the ones that are on the top, on the dorsal side of the brain. So these are the fibers representing the retinal quadrants, inferior visual field, versus Myers loop over here is superior visual field. So the Myers loop is a little bit more anterior versus the ones that are transmitting vision from the inferior visual field is going to be a little bit more dorsal. So D is going to be more of the Myers loop, which is going to be representing the superior visual field. If you wipe out that area, you're going to get left superior quadrantenopsia. So quadrantenopsia is one quadrant, basically, on both sides, right? So you're losing the quadrant. Then when you get towards the more posterior portion of the optic radiations, before you get to the primary visual cortex, if you wipe out that area, you're going to get a homonymous hemianopsia, but you're going to see macular sparing. So in other words, you're still going to have your central field of vision, but you're just going to lose both sides of the visual field. Okay, so just be familiar with these different types of visual field deficits. All right, other cell types that you're going to find. This is not super high yield. You have midget P cells and parasol M cells. Why is the midget P cell listed as a P cell versus the parasol as an M cell? You feel like it should be the opposite, right? It should be midget M cell, parasol P cell, but the nomenclature is flipped for whatever reason. The midget cells are really small. They're going to have a very small receptive field versus the parasol cells are going to have a much larger receptive field. The midget cells are going to be more receptive to color versus the parasol cells are going to not be as sensitive to color, but they do have a bigger receptive field in general. This is all FYI. I'm not going to test you guys on the different ganglion cells and their tracts. Same thing over here. I'm not going to test you on the different cell layers within the lateral geniculate nucleus. But just know that the geniculate nucleus in general is the region where you see a lot of processing of vision before it makes its way all the way back to the optic, the primary visual cortex. So here's a summary of your P cells, which are the midget cells, versus the M cells, which are your parasol cells. P cells, midget cells, are more sensitive to color versus your parasol M cells. Parasol because it's like a large, what do you call it, like a parachute, right? That's why it's called a parasol cell. You get lots of, it's very sensitive to a lot of information, so you can see low contrast stimulus. Right? It's kind of similar to your cones, right? Sorry, your rods. Your cones would be more similar to your midget cells. Rods would be more similar to your parasol cells. Now, again, everything is inverted, which we'll talk about here with the calcarine sulcus. The calcarine sulcus divides superior versus inferior. Above the calcarine sulcus is going to be your inferior visual fields versus below the calcarine sulcus is your superior visual fields, right? By the time it reaches the occipital lobe, the primary visual cortex, again, everything is upside down, right? But your brain basically just flips everything right side up. The fovea, remember that's where you have the highest concentration of cones, is highly represented by the primary visual cortex. Fovea is only 1% of the retina, but it is 50% of all the visual information being processed by the visual cortex. So in other words, image coming into the fovea is extremely, extremely important. It's one of the reasons why macular degeneration is such a horrible condition. You're losing a lot of your vision from that disease. The primary visual cortex is also referred to as the striate cortex, but for the sake of this class, I'm only going to refer to it as the primary visual cortex. Again, it's going to be in the occipital lobe. Again, the fovea is heavily represented, and the visual fields are inverted. So upper visual field is below the calcarine sulcus, lower visual field above the calcarine sulcus. The lateral geniculate nucleus, again, it's going to involve those parasol and midget cells. Right? The midget cells, color, parasol cells, very sensitive to light, so they pick up even like low light scenarios. Both of your eyes are going to be working together to allow for stereopsis. Stereopsis is binocular vision. So they're going to do something in this video which I think is kind of cool. Let's do it together. So with your hands far away, touch the tips of your fingers together. Okay. Now close one of your eyes and try to do the same thing. It's a little bit tougher. It's a little bit harder to do that. That's because with binocular or stereoscopic vision, you're able to see more depth, right? So you can actually see the depth of where your hands and your fingers are. So super cool stuff. Let's watch a little video on that, on stereopsis.
Stop. Pay attention. Put your hands to your sides like this. Now bring your fingertips up like this. Now, looking at your fingertips, touch them together. Easy enough, right? Okay, bring your hands back down your sides and close one eye. Now again, bring your fingertips up like this. And while staring at your fingertips, touch them together.
It's probably harder when you're drinking too, at the bar.
Not as easy as it looks, right?
If you couldn't touch your fingertips together with one eye closed, don't worry. You're not alone. With only one eye open, you weren't able to accurately gauge depth. Why is that?
I know, right?
In the right direction is Dr. Marchenke's. He's an expert in understanding how your brain perceives depth.
In reality, we don't see truly in 3D. It's a construct that our brain makes for us. The reason that we see it all is so that we can interact with the world around us, not bump into it. But the only way we could do those things is if we had perception of 3D. But there's a fundamental difficulty in perceiving in 3D. The retina at the back of your eye is two-dimensional. It's like a screen on which the world is projected. How can a brain take a two-dimensional input and figure out what that three-dimensional world is like?
To help answer that question, let's go bowling. Take a look at these pins. This is how your brain sees them with height, width, and depth. But neither of your eyes actually sees this image. This is what your left eye sees, and this is what your right eye sees. Your eyes have slightly different views of the same image, but your brain combines those views into one precise image of where the objects actually are in space, allowing you to throw a strike right down the middle. This is what scientists call stereoscopic vision.
The word stereo in your stereo system at home refers to the left and right speaker. And it's for the same reason that we use the word stereo when we refer to stereoscopic vision. When you're using only one eye to play this game of putting your fingers together, it becomes much more difficult because you've lost the stereoscopic depth effect.
To give you a better sense of how your brain uses stereoscopic vision to perceive 3D, take a look at this photo. Got it? Now here's another one. It's an image of the same trolley, but from a slightly different angle. Both look pretty two-dimensional, right? Do you think there's anything we could do to make them look 3D? How about this? We're not just shaking one image. We're actually quickly alternating between the two similar images. Doesn't it look like you could jump on the trolley and ride it all the way down the hill? Take a look at some more examples. Here are the two photos. And here's what they look like when we alternate them. Pretty cool, right? What you're looking at is a stereo animated illusion. All we're doing is jumping back and forth between two similar but different pictures. Your brain interprets the difference between the two flat images as depth, simulating what your eyes do with real three-dimensional objects. But stereoscopy is not the only tool your brain uses to turn 2D into 3D. Your brain uses other cues, like shading and size, to form a three-dimensional world.
So, cool stuff. All right, sorry. I'm not going to watch that one. This is FYI. These are the different layers in your cortex. Your cortex, for the most part, around your brain, you're going to have 6 layers in the cortex. The primary visual cortex is no exception. There are some exceptions to that. There's like some layers that, some regions that have like 4 layers, but again, I'm not going to test you on these different layers, the different cell types you find in the different regions of the cortex, which ones are ascending, which ones are descending, right? So that's, if you're interested in reading more into that, feel free to do so, but I'm not going to hold you guys responsible for that. Edge effect is another thing that your brain needs to interpret. It needs to interpret edges, it needs to interpret angles, right? So orientation of a stimulus to which the cortical neurons are maximally responsive. They've done this study using an anesthetized cat, and they measured the signals from the primary visual cortex to see which orientations are maximally stimulating, right? So for this, the orientation that looked like it was more like flat, straight on, was maximally stimulating for this cat in this experiment. So that's edge effect. So depending on the orientation, your brain is going to respond to different orientations, either more so or less so, depending on what the neurons prefer, I guess. This is another FYI. Neurons within cortical columns exhibit similar orientation preferences, right? So they were penetrating the different layers with electrodes and seeing what kind of effect that would have in terms of orientation, the orientation tuning curve, right? Which type of orientation of the vertical electrode would elicit a more cohesive type of firing pattern versus kind of a more random firing pattern in terms of spikes. So again, that's FYI. Yeah.
So the neurons like oriented in a way that when the signal hits them, they will relay the orientation.
Yeah, basically they operate. So they're in columns, right? So they're in those different layers. So within those layers, I think they're going to be communicating with each other in a certain way, right? And so they're going to have a preferential communication within those layers. Versus if you simulate them at an angle, they're not going to be coordinated as well together. Okay. So I think that's kind of the foundation of the results from this particular study. So. And then they all communicate with each other, right? Not each of those layers are going to give you the full picture. It's kind of like the analogy of a bunch of blind men touching an elephant in different parts, right? Whereas some of them will be touching the trunk and be like, all right, this is kind of like a weird like tree-like structure. The other ones will be touching the tail to be like, oh, it's just kind of this like rope-like structure, right? But they're not getting like the full picture of the actual elephant, right? The neurons in the brain are basically doing the same thing. Like this, for example, this is the complete picture in the middle here, the dog sitting next to this fence. Different regions in your cortex, in the visual cortex, are going to be creating a like part of the image. But then when you layer all those images together, then you get the complete picture. So that's kind of what's going on with edge detection in the cortex. All right. Some other things to cover. Again, this is not going to be super high yield. I'm not going to test you guys on this, but there's the ventral and dorsal streams. The ventral stream, which is like on the the lower portion, right, versus the dorsal portion, which is above. The ventral stream is going to be communicating with the temporal cortex, and it allows for object recognition, versus the dorsal stream that's on top is going to be communicating with the medial temporal area, which is going to be allowing for more spatial interpretation. So determining what three-dimensional space looks like. But I'm not going to hold you guys responsible for that one. So ventral pathway, temporal lobe. Dorsal pathway is eventually going to lead to the parietal lobe, which allows for more of your spatial recognition. This, however, is something that you are going to be responsible for. So pupillary dilation and the light reflex, right? Autonomic activation, right? This is part of the autonomic nervous system, both sympathetic and parasympathetic nervous system. The region in the brainstem that's going to be responsible for cranial nerve 3 is going to be the Edinger-Westphal nucleus. So Edinger-Westphal nucleus is going to be allowing for your ocular motor nerve to operate. Parasympathetic stimulation, when you're just chilling, you're not doing anything, maybe you're in rest and digest mode, maybe you're sleeping, your pupils are going to be constricted. Now, if you're in a fight and flight mode with sympathetic activation, you have to see your surroundings, right? Like if you're fighting off a predator or something like that, you want to see as much as possible. So you're going to see more pupillary dilation. Again, cranial nerve 3, Edinger-Westphal nucleus. That's going to be the main regions associated with the pupillary constriction and dilation. So here's what's going on with the light reflex. So here's the steps. First, you have the bilateral projections from the retina to this region called the pretectum, and then that's going to eventually communicate with the Edinger-Westphal nucleus. Then you're going to see neurons from the Edinger-Westphal nucleus going back towards the retina, and then that's going to communicate with the ciliary ganglia, and that is what allows for the pupillary constriction muscles to be activated. So light information going through cranial nerve 2, right, the optic nerve, making its way to the lateral geniculate nucleus, which eventually makes its way to the Edinger-Westphal nucleus within the brainstem, is going to now send signals back to the eye, right, to the ciliary ganglia, allowing for the pupils to constrict, right, so the pupillary constriction muscles. Now, the other thing that we talked about earlier on this semester was the accommodation reflex. Or the accommodation reflex is when something is close versus far away. Right? When you have the patient look at their finger up close versus looking at an object far away. So that's also going to involve the ciliary muscles. It's also going to involve the Edinger-Westphal nucleus. When something is close, you're going to see accommodation, convergence, and constriction. Right? Your pupils constrict. Your eyes are going to converge. You're going to look a little bit more cross-eyed, and you're going to see an accommodation reflex, which is going to actually change the shape of the lens of the eye so that you can focus close in. Another thing that you see happening in the eye is your eye movements. So you have conjugate eye movements so that the eyes can move together, right? The extraocular muscles of the eyes have to be able to operate in synchronicity to be able to allow for conjugate eye movements to take place. If you have any sort of disorder with this process, that's going to be resulting in nystagmus. Nystagmus can happen in many conditions. It can happen with certain diseases, like it could happen with like vertigo, for example. There's benign positional vertigo, depending on the patient's position. If they're laying down versus upright, they might start getting vertigo, and that can cause the eyes to start moving as well, because you start like losing balance of the world. Vertigo is like when the whole entire world is basically spinning, so that's going to affect your eyes. But there's also drugs can cause this too, like alcohol famously causes nystagmus, right? If you get pulled over and the cop suspects that you're drinking, they're going to do the eye test. They're going to see your eye tracking, and when they go laterally, usually if you're sober, your eye is going to remain focused and fixated on the finger. If you're drunk and this process is not, the convergence is not working properly, then your eye is going to be going back and forth, nystagmus. Right. So let's watch a little video on nystagmus.
Thank you very much for coming. Appreciate it an awful lot. Derek has had MS for a few years and he's on beta-ferron and doing reasonably well, but he has an unusual eye movement disorder that is usually pathognomonic of MS called internuclear ophthalmoplegia. So when you're examining someone with MS, you look specifically at every examination. So Derek, if you don't mind, can you look at my finger here? So rest once more. There's no evidence of ptosis. No square wave jerks, and the pupils are equal. So if I can get you to keep your head still, please, Derek, and look to your left. So we've got some nystagmus on lateral gaze there, as you can see. More marked in the left eye than the right, which is the big hint here as to whether there's an underlying right-sided INO looking to the left. But much more obviously, can you look down to your right? He's got very marked nystagmus in his right eye with a failure of adduction of the left eye. Left, right, left, right, left, right, left, right, and back in. So he's actually got a bilateral internuclear ophthalmoplegia due to MS plaques in the MLF, or median longitudinal fasciculus, in the brainstem, connecting the third nerve in the midbrain and the sixth nerve in the pons.
So in other words, you can use nystagmus and a physical exam to actually be able to, like he did, isolate where the lesion is taking place. So lateral geniculate nucleus for that patient. So super interesting stuff. The major types of movements are going to be smooth movements versus saccadic movements. The smooth, as the name implies, smooth, right? Saccadic is going to be rapid, sudden change, right? So that's what's going on over here with this guy. So this is smooth pursuit versus more of like a saccadic type movement, which would be more characteristic of nystagmus. Saccadic suppression, right? That's going to be a way for your vision to become more precise, right? It reduces the blurring of an object. So let's do a couple quizzes questions before we get into the next topic. Oh, geez, they changed it again. They keep on changing quizzes. I think it's instructor pace is what I usually do. I hope. So go ahead and sign in. We'll do like one or two because I really do want to get into the next topic of hearing. All right, let's go ahead and start. All right, cool. 70-year-old female. She goes to the ophthalmology clinic. Distortion of her central vision. Difficulty reading, recognizing faces. Fundoscope would reveal what? First of all, what condition is this? Yeah, this is macular degeneration. With macular degeneration, you're going to see, especially with dry macular degeneration, you're going to see drusen deposits. You can see also pigment changes, atrophic changes. Something else that you might see as well if it was wet macular degeneration would be hyper or neovascularization in that area. It's called wet because you're getting more blood vessels, and those blood vessels are more prone to rupture. This would not be the answer. Optic disc swelling, right? That's where you don't have vision, right? The optic disc. Right? So this would be in the macula, particularly affecting the fovea as well. All right, let's do one more. So that should be a softball question. All right, synesthesia. Super easy. Yeah, that's not dyslexia. It's synesthesia. All right, ending it there. Make sure you do the rest of those because there's quite a few questions on those retinogeniculocortical tract pathologies towards the end. You definitely want to be familiar with those. Any questions on vision before we move on?
No, but is it a thing where people's blue eyes are more sensitive to like brighter?
I actually don't know that. I don't know. Let me look that up. Generally speaking, patients with lighter colored irises, such as blue or gray, experience more light sensitivity than someone with brown eyes. The density of pigment in light eyes is less than that of darker colored irises, right? Because that's due to pigmentation, like melanin, right? And when the light hits the dark colored iris, the higher density pigment blocks the light rays. So yeah, absolutely. It is interesting. You guys are asking great questions today. I love it.